Okay. All right. I am here with Dr. Laura Lagomarsino. And can you start out by telling us what your job is?
Yeah, thanks for inviting me to talk with you today. I am a new assistant professor and herbarium director at Louisiana State University. And so I both run a lab and teach courses as an assistant professor, but as the herbarium director, I'm in charge of a scientific collection of plants. I have about 400,000 plant specimens that you can use in scientific research, and I'm in charge of keeping them intact.
Awesome. So I'm guessing some of my students probably have no idea what an herbarium is. So you sort of just described it, but yeah.
Yeah, so the herbarium is kind of like a library of plants. They are pressed and dried plant specimens, so flattened, that are on sheets with information about where this plant was collected. So for example, lots of botanists will go out to the field and collect a bunch of plants from a single ecosystem, like a wetland here in Louisiana, and bring them back. And so the herbarium sheets have information about when and where this was collected, the time of year, the collector, and the herbarium serves as a repository for plant diversity through space and time. So any researcher on campus can come, and outside of LSU can come use this to see how the plant diversity has changed or what different species look like, to categorize new species. And at LSU and at lots of institutions as well, the herbarium is complemented by a museum that has vertebrates and invertebrate collections as well, and sometimes even rocks and fossils. So the herbarium, though, is the plant, and lichen and fungus complement to those collections.
Right.
So
all of my students know the difference between plants and fungi by now.
Yeah. I hope so.
Cool. So balancing, so teaching, researching, managing managing the herbarium. I'm sure there is no typical day, but what is a typical day, or maybe it's easier to talk about cycling through a semester a year. What does it look like for you?
Yeah, I started this job very recently in mid-August, so I don't have a typical anything yet. Everything's new and changing, but I anticipate that a typical day will be a combination of responding to emails and concerns related to the herbarium, spending some time in the classroom or lab teaching, and then also doing my own scholarship, writing up my research and doing data analysis, some data collection. And one thing that I've realized in becoming a new professor is that my previous jobs as a postdoctoral fellow and before that as a graduate student, I was in charge of my own research project. But as a professor, I am now in charge of helping people do their own projects, whether that is my postdoctoral fellow working on her research project, which will be peer-reviewed and published down the line, or my collections manager in the herbarium identifying problems that we need to find solutions for in the herbarium. So a lot of my job is becoming sort of the taskmaster of many tasks at once, becoming a multitasker. But the days will change. Some semesters I'll be mainly teaching. Some semesters it's going to be big herbarium crunch to accommodate new collections that we've gotten. But I think in general, it's the delegation of tasks that's going to make my life possible in balancing all these different tasks.
Managing all these other things. Awesome.
Exactly.
Let's back up a little bit. When did you first know that you wanted to be a biologist?
Yeah, I was always interested in natural history, being outside. My parents camped with me a lot, so we spent a lot of time in the forests, the redwood forests of Northern California. And so I knew I wanted to do something related to ecology or being outdoors. I didn't know it was specifically plants, and I didn't know it was specifically evolution and taxonomy, with the sort of my fields now. But I would say from a kid, I knew I wanted to be a biologist, or at least do something outdoors with plants and nature. And it wasn't really until I was an undergrad doing undergraduate research that I realized that I wanted to be a the sort of biologist I am today.
Okay, so that was you were what type of research were you doing as an undergrad? Were you working in a lab?
Yeah, so I was an undergraduate at UC Berkeley, and I worked in a lab. It was a brand new lab, just like the lab I'm starting now, that worked on tropical plant systematics. So systematic being the inference of relationships between different species and how to classify these groups. And I really fell in love with the plant group we worked on. These are cool tropical group of plants, and I really wanted to go to the rainforest. But I also got really fantastic mentorship and from my undergraduate advisor, and a lot of experience in diverse projects, and that sort of just been down the same sort of trajectory ever since then. So it's been a
Cool.
very straight line for me in terms of research experiences.
Then
Yeah.
did you go straight from undergrad to grad school?
I took a year off between undergraduate and grad school. I, but I wouldn't, I was still a student in a lot of ways.
Yeah.
So I studied abroad. I took a course through the Organization through Tropical Studies on tropical biology.
Okay.
And that was a semester-long course.
Yeah.
So I finished my undergraduate degree, but still kept on taking undergraduate classes. And then for the other half of the year, I worked as an intern at the Smithsonian. So the Natural History Museum at the Smithsonian is famous for its big elephant in the rotunda and other amazing public exhibits. But if you go behind the scenes, they have an herbarium, they have a museum with birds, a museum with insects, museum with fossils, a museum with minerals. So I spent 6 months working there before starting grad school at Harvard across the river.
Where we met. Great.
That's where we met. Yep.
All right. So while we're in sort of undergrad world, do you have any advice for undergraduates? Most of my students, most of them, not all, are pre-med, but just sort of broad advice for a young person starting out thinking about different career options in biology broadly.
Yeah. I think that if you're interested in biology, the best way to get a sense of what the career will actually look like is to get as much hands-on experience as you can. If you're interested in academic science, like what I do, I would say getting into a research lab is really important. A lot of times, professors are looking for undergraduates to do some basic research, and if you're able to find a position where you're doing not just sort of maintenance of the lab, but doing research where you're asking a question and a graduate student or postdoctoral fellow is helping you guide research, I think that's the best way to really learn how this sort of science works.
Yeah.
But there are also internships at museums, like I mentioned, Smithsonian or other places like that, or the USDA if you're interested in applied plant biology, that are often looking for, especially summer interns, and that would be another option.
That's a
Um,
great idea. I'm glad you mentioned that. I think there's a lot of bio majors at BC, and it can be really hard to get into the labs of the biology faculty there. So
yeah.
other options like that are great for students to think about.
Yeah, definitely. And if you're interested in those, the National Science Foundation has a research experience for undergraduates program that's really worthwhile looking into.
Yeah.
And a lot of the places that draw on undergraduates are universities, sometimes big, sometimes small, but they're also other types of institutions, like I mentioned.
Cool. That's great. Okay, so let's talk a little bit about plants. You're a plant biologist. We've just learned everything there is to know about plants in 2 classes worth. So clearly we're experts. What do you think are the big unanswered questions in the world of plant biology that would most excite you to have answers to in the future?
Yeah. So let me see. That's a really big question. And my biology is really narrow.
Well, then tell us about your sub. You feel free to narrow that into whatever subcategory you want.
For sure. In terms of questions I'm excited about in plant biology, so I'm an evolutionary biologist who's really interested in sort of the broad pattern of how species are related to one another. And one of the big questions in my field, and this is shared across sort of systematists outside of plant biology as well, but is how old the different plant groups we study are.
Yep.
And so the angiosperms, or flowering plants, so these are the plants that produce flowers and fruits, are one of the biggest, they are the biggest major group of plants around, and they're known as Darwin's abominable mystery. Not because they got as diverse as they are, but because there's a, in the fossil record, they go from having no record of their pollen in the fossil record. So how we often track plant origin through time is looking at the little parts of the plant, spores of ferns or pollen of gymnosperms, so conifers, or angiosperms, or flowering plants. And when you look at that fossil record for angiosperms, it goes from nothing to everything in a very short span of time.
Yep.
And when you infer relationships of flowering plants and use the molecular rates of mutation and you try to infer ages with that, you get a very old age for angiosperms stretching back 100 million years earlier than when we first see their pollen arise.
Okay.
So for me right now, in my subfield, what I am most excited to see happen is for new ways, new methods to look at these data to maybe reconcile the fossil record and the molecular dates a little bit better, because it's a huge discrepancy right now. And of course, when angiosperms arose has a lot to do when the insects that eat angiosperm leaves evolved, when frugivores like toucans or monkeys evolved. So there are just so many questions that rely on that, and there's a lot of hand waving right now. And so we can know more about that either by looking into the fossil record more deeply, trying to find hidden gems going further back in time that we just haven't seen yet, or by improving our computational methods of exploring molecular age estimates.
And that would all be based on DNA sequence data, is that correct?
Exactly, on DNA sequence data, and sometimes a combination of DNA sequence data with what we know about the age of different fossils.
Okay, great.
So that's,
so we've talked a lot. We've talked a lot about evolution's internal consistency and how when the fossils match up with the DNA, matching up with the morphology, allows us to be really, really confident. But it sounds like you're talking about something that I was not aware of in the plants, is that there's this really interesting discrepancy between what we know about the fossil record and what molecular phylogenies are telling me, and that you and other people in your field are trying to get more evidence to figure that mystery out.
Exactly.
Cool.
Sort of solve Darwin's abominable mystery. Exactly.
Awesome. Cool. Do you have a favorite plant?
Yeah, it's hard for me. I love a lot of different plants.
Yeah.
And the plant group I study, I have a soft spot in my heart for. So I study neotropical, so American tropical lobelia relatives. So lobelias are the little purple plants that you can get at Home Depot that are really common in hanging baskets in Boston in the summer. But I study their tropical relatives, and I love them. I've described new species and have learned a lot about their evolution through my research. But I think my favorite species, as I sort of alluded to earlier, is the coast redwood of California. Yeah. So these are the plants that got me into biology, and they are beautiful, gigantic trees, the world's tallest trees, over 300 feet tall, and they're just special to me. So
awesome.
They're my favorite.
Cool. All right, so I think it's easy for undergraduates to see successful professionals and have trouble envisioning themselves at getting to that stage. Would you be willing to share either a failure story or some challenge that you've overcome in your career trajectory?
Yeah. So I would say that I I think it's really difficult for people to see themselves in science a lot of times because representation in science doesn't match representation of populations generally. Um, I've had a fairly straight shot experience in science to date, but I would say a lot of that's because I've had really good mentorship in my past, particularly from my undergraduate advisor, who was a woman whose lab was full of female graduate students and full of female
Okay.
postdoctoral fellows, and she has made sure that her lab is balanced, and she's now a leader in my subfield of botany, and she's a huge voice in reminding us that we need to have diversity of people represented and at the highest levels. And I think in botany we tend to think we're pretty good at gender representation.
Yep.
There are many more senior females than in other subfields.
Okay.
But even in botany, we do a really poor job at the highest level of having gender equality represented. And I think a really big example of that happened about a year ago when the International Botanical Congress, which is a really big conference for my subfield that happens once every 6 years, they announced their chosen keynote speakers, and there were 40 of them, and 3 of them were female. And that's sometimes very typical in science.
Yeah.
And I was really happy to see. Oh, and talking about sort of other diversity, they were all white or Chinese, and this meeting was in China, and so there wasn't great racial diversity represented either.
Yep.
And I was really happy to see my community get really upset about this in a public forum on Twitter, on Facebook, and then eventually I learned through emails to the leaders of this conference to make sure that this was addressed. And through this push by the community, we made it. It wasn't equal. It never reached 50% female, male.
But it got better.
Diversity wasn't represented, but they added many more speakers, and so females were represented. So I think a lot of the idea of white men as being the scientists as being really challenged within the community, but there's still a lot of active pushback that needs to happen, even from within our own subfields that think they're pretty good.
Yep.
So yeah, so that's my experience, and I know that a lot of women and a lot of people in color feel this way in science. It's getting better. It's better than it was.
Yeah. Great. Thank you for sharing that. I had a question that I probably should have asked you earlier when we were talking about collections, but I know you thought a lot about the importance of collections beyond just plant collections to biology. This is something we haven't really talked about in my class, so I'd love if you could talk just for a little bit about why you think these collections that a lot of people don't even know exist and that are some of the threats to these collections and why they're so important.
Yeah. So these plant collections, as I mentioned earlier, are a record of plant occurrence and plant phenology. So whether plants, the temporal changes in a plant, so going to flower, going to fruit, losing leaves. So they're a record of these traits through time. And I think that that's always important. We want to be able to predict when something should be in flower. We want to know how appearance of a plant has maybe changed. The most traditional uses of these collections have to do in describing new species and looking at the species that occur in a specific area, just writing what is called a flora. But I think there are a lot of new uses for collections that are just in the last maybe decade or decade and a half have started to be explored. And I think the power of these new approaches really comes in looking at changes through time. So especially in regards to some of the anthropogenic changes that are happening, such as climate change. We know that temperature is warming. And my favorite example of using collections-based data to look at changes through time in a collection was actually by my PhD advisor at Harvard and Richard Premack. So my PhD advisors, Chuck Davis and Richard Premack, who I think is at Boston University.
I believe so, yeah.
Yeah, they use the collections at Harvard that were made by Thoreau and people who came after Thoreau. So Thoreau is really good at documenting when plants came into flower and in the spring. So he'd go out every spring consistently to see when buttercups came into flower, when roses, when violets, etc. And people, there are two really big data sets of people who came after him who also did that.
Okay.
And we know that from Thoreau towards the present, spring has been starting earlier and earlier in the Boston area. And we want to see what that effect has been on these communities of plants out in Walden Pond, which is where he was doing this research. And so they use these herbarium collections made by Thoreau and the people who followed to show that plants have sometimes shifted their phenology. Some groups are doing a really good job of flowering earlier with the spring, but other groups of plants, including a lot of the ones that are blooming later, that are conservation interests, things like irises and orchids, they don't shift their phenology. They don't flower later. And so there's a mismatch between when pollinators are present and when flowers are opening.
Got it.
And an environmental mismatch. They should be flowering when it's cooler.
Yeah.
And then later shifting to when photosynthesis is more efficient, but they're not. So
Yes.
I think that's a really good example. And there are tons of others like that, looking at coal pollution through time, or looking at water contamination with heavy metals in the leaves. So there's a lot that you can learn using these collections, especially if you see it as sort of a record of change through time.
Yeah.
Yeah.
Great. Thanks. I think that was everything that I specifically wanted to ask you about. If I have any students that want to follow up with you or follow your research or ask you specific questions, what's the best way for them to do that?
Yeah, you can either come find me on Twitter. My handle is Lagomarsino_L. Not as great. So Dr. Olins can give you that info. Or find all my information on my lab's website, which is lauralago.net.
Great.
Yeah, and I'm happy to respond to any emails you send my way.
Awesome. Well, thank you so much. I really appreciate you taking the time to talk with me and talk with my students.
Oh, thank you so much.